Hello, and welcome to today's webinar. I'm Phillip Byrne, and together with Dr. Aimee Silla, run the Evolution and Assisted Reproduction Laboratory at the University of Wollongong, Australia. In this webinar, myself and Dr. Silla will be discussing the use of artificial fertilisation in the genetic management of threatened anurine amphibians, with a focus on managing genetic variation. So what is artificial fertilisation? Essentially, AF is a technology that is increasingly being used to propagate endangered amphibians and involves the collection of gametes from both sexes that are then combined under standardised conditions to generate offspring. Critically, AF allows direct control over parentage and the generation of offspring of known genetic history, providing a very powerful tool for managing the genetic composition of captive and wild populations and preserving genetic variation, a goal that is endorsed by the IUCN. So why is there such a push to maintain and preserve genetic variation? The hard, fast answer is that loss of genetic variation represents a major threat to the persistence of populations, with irrefutable links between the loss of genetic variation and the extirpation of populations. When populations become smaller and genetically isolated, the inevitable outcome is that they will lose genetic diversity through genetic drift and directional selection. In turn, with each passing generation, they will become increasingly inbred and more susceptible to a loss of fitness, a phenomenon we term inbreeding depression. This problem will be compounded by reduced adaptive potential, which is expected to elevate the risk of extinction, particularly in environments exposed to rapid environmental change. In these situations, it is essential for conservation managers to develop effective strategies to preserve genetic variation. So just how do we maintain or increase genetic variation in declining populations? Arguably, the simplest way to achieve this is through population augmentation. Population augmentation, also referred to as supplementation or restoration, involves supplementing a declining population with conspecific individuals from one or more genetically distinct source populations. So how does population augmentation provide genetic benefits? Well, in principle, providing a declining population with new genes, or more specifically new alleles, sourced from one or more populations with higher genetic diversity, stands to overcome the problem of inbreeding depression. Essentially, the integration of novel genes is really expected to counter the expression and accumulation of deleterious recessive alleles. In turn, this should improve the recipient population's resilience and persistence by reducing vulnerability to demographic or environmental stochasticity. These presumed evolutionary advantages are termed genetic rescue and have led to population augmentation being widely recommended as a conservation action. Genetic rescue harnesses the phenomenon of heterosis, which is the observed increase in vigour and progeny from matings between genetically different strains or populations, a phenomenon that is also commonly referred to as hybrid vigour. Theoretically, there are three main reasons for hybrid vigour. The first reason is dominance. The general notion here is that crossing brings together dominant genes that are more favourable for vigour than recessive genes. Essentially, dominant genes from one parent may complement the dominant genes from another parent. Stated simply, this explanation hinges on the idea that dominant genes are better for viability than recessive genes. The second reason for hybrid vigour is overdominance. The general principle here is that heterozygosity is superior to homozygosity because different alleles produce different yet favourable effects, whereby the combination of effects in a heterozygote is beneficial. Stated simply, more genetic variation overall is much better for the viability of offspring. The third reason for hybrid vigour is epistasis. Here, the principle is that interactions between alleles on different loci have positive effects on individual performance and viability. Stated simply, more interactions between a greater number of alleles is better for offspring viability. Despite hybrid vigour being widely demonstrated in nature, there's actually a growing concern that mixing populations might not always provide beneficial outcomes. More specifically, there is concern over the risk of long-term negative genetic consequences resulting from matings between individuals from evolutionary distinct populations, a phenomenon we term outbreeding depression. So what actually causes outbreeding depression? There are two main reasons. The first is that parental populations are adapted to different environmental conditions, and hybrids possess intermediate phenotypes that make them less fit in one or both environments. This phenomenon is termed extrinsic outbreeding depression. The second reason is that matings between locally adapted populations can result in the disruption of co-adapted gene complexes, leading to genetic incompatibility, reduced fitness of the offspring, and a compromised population viability, a phenomenon that is termed intrinsic outbreeding depression. So do amphibians face a high risk of outbreeding depression? The answer is most definitely yes. This is largely because amphibians typically show an extreme level of genetic differentiation between populations due to strong philopatry. Many amphibians return to the same site to breed year after year. Due to low dispersal abilities, for many species, tadpoles are stuck in disconnected ponds, and small metamorphs rarely move long distances very quickly. And it's also due to the uneven distribution of breeding habitats. Typically, what we see is places for anurans to breed are very scattered in the landscape. Consequently, for any given anuran species, there's an inherent risk of outbreeding depression that necessitates empirical assessment of the genetic consequences of mixing populations before a program of population augmentation is initiated. Fortunately, artificial fertilisation can come to the rescue here, as it provides an excellent tool for testing the genetic outcomes of mixing populations and can be used to address and potentially alleviate concerns over outbreeding depression. For species characterised by external fertilisation, a direct way to test the feasibility of population augmentation as a conservation action is to use artificial fertilisation to perform controlled breeding experiments that directly test the genetic consequences of mixing different genetic lines, different populations. How are these breeding experiments conducted? Well, put simply, gametes are obtained from both sexes, and AF is used to make crosses between individuals from donor and recipient populations that are being considered for use in a program of population augmentation. Subsequent assays for the fitness of the progeny from each family that is generated then allows direct evaluation of the genetic consequences of outbreeding. Anurans are really ideal for this type of investigation because, for many species, large numbers of gametes can be obtained from both sexes, allowing for comparison between tens of individuals across multiple populations. So while these experiments are conceptually simple, there are a number of experimental details that require careful consideration. Why? Well, the reason is that poorly designed studies can generate spurious findings that misdirect management practices. In this webinar, we provide recommendations for running breeding experiments, and we outline an experimental design and a set of methodologies that allow an effective test of the feasibility of augmenting populations. To kick things off, let's ask the question, what is the best breeding design to use? Well, without question, the most balanced and systematic biometric approach to deduce the combining ability of populations that have been earmarked for an augmentation program is to employ a full diallel cross mating design, which you can see pictured here. In this context, the design, which is also often referred to as a North Carolina type 2 mating design, involves taking a set of males and females, ideally in equal numbers, but you can also use uneven numbers, as Aimee will outline in our case study later in this webinar, from 2 or more genetically distinct populations. Here we're considering 2 populations, population A and population B, and then crossing every parent, every genotype, in every possible pairwise combination to generate both purebred families, which are highlighted in the bold cells, as well as hybrid families, which are highlighted in the grey cells, from all possible parental genotypes. Critically, the full design should also include reciprocal crosses, whereby crosses are made between females from population 1 and males from population 2, as well as between females from population 2 and males from population 1. These reciprocal crosses make it possible to test for population-specific paternal and maternal effects. That is, the genetic influences coming from mum and genetic influences coming from dad. Understanding the relative importance of these effects may be critical in anurans, given the findings of a breeding experiment which reported that estimates of outbreeding depression in European common frogs were strongly influenced by which sex and population was tested. So just to reiterate, you can't just take sperm from one population and eggs from another population. You need to take sperm and eggs from both populations and mix them in every possible pairwise combination. This is critical because the degree of hybrid vigour can depend on whether the DNA comes from male or a female. If you only use males from population A and females from population B, you're likely to miss important maternal or paternal contributions to offspring viability. Importantly, the breeding experiment can involve animals from more than 2 populations. For example, you could make crosses between frogs from 5 different populations. In the example here, you can see a cross between 5 males, representing populations A, B, C, D, and E, and 5 females from the same 5 populations. Again, you need to make reciprocal crosses so that you don't just have sperm coming from one population and eggs coming from a different population. You need every male to produce offspring with every female and vice versa. So while this design is powerful, there's an inherent risk of introducing temporal confounds. Crosses need to be performed quickly after collecting the animals and gametes. Otherwise, gamete quality can decline quickly, which can influence fertilisation capacity and offspring performance. This limits how many animals you can work with at once. To overcome this challenge, we recommend that the full diallel cross design is used in combination with a randomised block design. So what is a randomized block design? Well, essentially the diallel cross experiment that we're talking about here is replicated over several experimental blocks. That is, the entire experiment is repeated multiple times. For example, here you can see the experiment is repeated 3 times, represented by the 3 experimental blocks, block 1, block 2, and block 3. Critically, each new block must use a new set of randomly sampled individuals to avoid the problem of pseudo-replication, where some samples are not independent of each other, a cardinal sin in experimental design. So just remember, use new frogs in each block. When designing an experiment, another important question to ask is how many parents should be used. Unfortunately, there is no hard, fast, correct answer here. It really does depend on the information being sought. However, a good rule of thumb is that a minimum of 4 parental crosses are needed to allow some basic genetic insights to be gained. Reflecting this, if you take a look at the literature, the number of parents used typically ranges between 4 and 20. Another important question when designing your study is to ask how many offspring should be tested per family. Again, there is no hard, fast answer as it really depends on the information being sought. Saying this, using very low numbers of individuals, say 1 or 2 eggs per family, would greatly increase the risk of generating imprecise and spurious estimates of mean fitness. Perhaps the most logical approach is to maximize sample size based on biological and practical constraints. For most anuran species, clutch sizes are large, greater than about 100 eggs. So in principle, it should be possible to have at least 10 to 20 offspring per family in a diallel experiment involving 5 females. This is a good sample size that is likely to generate meaningful data and provide some important insights into the risk of outbreeding depression or the likelihood of heterosis, hybrid vigour. Let's now move on to another very important question, and that is what generation should be tested? Here, there is actually a very clear answer. Ultimately, you'll run crosses all the way through into the 3rd generation. I'll come back to this in a minute. Without question, looking at just one generation, the F1 generation, the 1st generation, can provide a pretty good and quick insight into whether populations are compatible or not. If eggs in many of the hybrid crosses are failing to fertilize, or you're seeing a high proportion of malformed offspring, it's highly likely that you're observing signs of genetic incompatibility and outbreeding depression. So a lot of information can actually be gained quickly from crossing for just one generation. And this would most certainly be much more informative than trying to predict whether outbreeding depression will occur, for example, based on the degree of genetic differentiation between populations. Saying this, in theory, reductions on fitness associated with outbreeding depression are expected to be manifest in the F1 generation and then become more evident in the F2 generation and beyond. As such, we should really test for outbreeding depression into the second and third generation. What causes the reduction in fitness in the second generation, the F2 generation and beyond? There are two reasons. The first reason is that between population heterozygosity that increased in F1 in the first generation and may have actually caused some hybrid vigour being observed, will effectively be halved by the process of segregation into the second generation. The second reason is that recombination that occurs in the F2 generation and beyond may progressively disrupt locally adapted combinations of parental genes that are present in each of the source populations. So moving on, the final question we need to ask is, what types of analyses should be done? The simplest analyses you can do is to make comparisons between the viability of individuals in purebred crosses, shown here in the black cells, versus hybrid crosses, shown in the grey cells. Here, standard statistical analysis can be used, such as linear mixed effect models, where you have cross type, purebred or hybrid, as a fixed factor in the model, block as a random effect, and the fitness measure of interest, for example, fertilisation success or maybe offspring body size, as the response variable. This is a common analysis that can be done by anyone with a basic understanding of statistics. Most biologists should be able to run this for you, or at least show you how to do it. If you want to get a bit more serious, you can also undertake some more sophisticated analysis that test for hybrid vigour. And there are two common tests used. The first tests the differences in the performance of the hybrid relative to the average performance of the parental line. And this is termed mid-parent heterosis. The second tests the difference in the performance of the hybrid relative to the best parental line. And this is termed better parental heterosis. If the outcome of these tests is that the hybrid line has higher value than the purebred parents, there is evidence for heterosis, for hybrid vigour. But if the hybrid has lower value, you have evidence for outbreeding depression. So anyone with some basic math training should be able to do these tests, as the math behind these calculations really isn't hard. Another thing you can explore is combining ability, defined as a population's or a genotype's ability to combine with other populations or other genotypes during the hybridization process. Two types of combining ability are measured. The first is general combining ability, GCA, which measures the average performance of a parental genotype or line in a series of hybrid cross combinations. The measure identifies the lines that generally lead to high offspring performance over different line crosses. Ultimately, this measure really helps us identify lines that are generally good for crossing. A more specific measure of combining ability is specific combining ability, or SCA, which measures a specific combining ability of two lines or two parents that you're interested in. That is the performance of a parental genotype in one specific hybrid cross. What this measure really does is to identify cases where certain crosses do relatively better or worse than expected on the basis of the average performance of the lines or populations involved in multiple cross combinations. That is, the measure really considers the deviation of the one cross that you're interested in relative to the GCA. So if we break it down, what exactly do these measures really tell us? Well, GCA and SCA provide information on gene action involved in the expression of quantitative characters, the fitness determining traits that are being measured, such as fertilization success or body size. The measure allows identification of generally good genotypes or populations for breeding. This is the GCA, or genetically compatible or incompatible genotypes or populations. This is the SCA, the information that you get from just looking at the one cross combination. So essentially, the SCA is used to identify a cross that can be used to exploit heterosis to improve population viability. So who has the expertise to perform these analyses? Well, the answer is most quantitative geneticists. Calculating combining ability is widely used in plant and animal breeding, and there exists an excellent knowledge base and set of resources that can be utilised for amphibian conservation breeding programs. But to make your lives easier, we would strongly recommend collaborating with a quantitative geneticist. One can likely be found in any good university near you. Anyway, the take-home message from all of this is that analysis of combining ability allows practitioners to identify suitable parents for hybridization and suitable cross combinations that may be used to exploit heterosis, hybrid vigor, to improve a population's viability through augmentation. But please remember that the extent of heterosis will be trait dependent. You need to measure traits that will have a direct and significant impact on fitness. Aimee will now talk about this more in a few minutes. So after you run your experiments, what possible outcomes and responses might you see? At one end of the spectrum, you may get evidence for moderate to high levels of genetic incompatibility and low heterosis. If this is the case, there's a real risk that mixing populations could compromise the viability of a target population and accelerate its decline. In response, managers should seek alternative management strategies. At the other end of the spectrum, if the experiment reveals low to moderate levels of genetic incompatibility, there is a low to medium risk of outbreeding depression, and managers should weigh the risk of outbreeding depression against the immediate risk of the population going extinct using a structured decision-making framework. If intervention is essential and in situ management is unsuitable, the best approach may be to introduce a relatively small number of individuals each breeding season. Just how many foreign animals could be introduced? In theory, if crosses are not 100% incompatible and the level of gene flow from the donor population doesn't exceed 20%, incremental introgression of new genes may lead to the recovery of population fitness without compromising locally adapted gene complexes. Under this scenario, artificial fertilization could be used to screen for incompatibilities, with the subsequent release of animals restricted just to those animals that are the most viable. I'll now hand you over to Aimee.
Thank you, Philip, and hello, everyone. I am Aimee Silla, and I will spend the remainder of this webinar drawing your attention to a few practical issues that should be considered when conducting artificial fertilizations and rearing offspring, specifically for the purpose of testing genetic outcomes between crosses. I will also identify the critical fitness determining traits to measure throughout different amphibian life stages, and we'll finish with a brief case study discussing the results of a research project that Philip and I conducted in one of Australia's terrestrial toadlet species. First, gametes need to be obtained. This can be done either post-mortem or non-invasively via the administration of reproductive hormones. Hormone therapies used to induce gamete release have been discussed previously in webinar 7, and so I won't cover them again here. I will draw your attention to a recent book as well as some review articles listed here for further reading on this topic of hormonally inducing gamete release and gamete collection in amphibians. When conducting artificial fertilizations, it is essential that gametes of maximum viability are collected and that viability of gametes does not vary among crosses. Importantly, when conducting fertilizations for the purpose of testing genetic outcomes, females that are hormonally induced to ovulate must be stripped of eggs, as seen in this video here, and fertilizations should be conducted immediately. Spontaneously ovulated oocytes should not be used, and oocytes should not be stored prior to fertilization, as the oocyte's capacity for fertilization declines within minutes of oviposition. Such declines will result in variability in the fertilization rate, of oocytes between your females, which will falsely inflate maternal effects. For this reason, it is also recommended that more females than are required to complete your full suite of crosses are induced to ovulate simultaneously to ensure that clutches of oocytes can be collected from females and subsequently fertilized in very quick succession. Any females that exhibit a delayed ovulatory response should be excluded from these type of experimental studies. To avoid potential differences in the capacity for fertilization between the first and last oocytes expelled from each female, it is important to ensure each subclutch has a mixture of early and late oocytes. In principle, this can be achieved by systematically stripping a small number of eggs into each fertilization tray and repeating the process until the female is spent. Alternatively, an entire clutch of eggs can be stripped, gently mixed, and then randomly allocated to your fertilization trays. Sperm suspensions retain viability longer than oocytes once collected, and for this reason, suspensions of sperm should be pre-prepared minutes to hours in advance and placed in cold storage, either refrigerated or on ice until use. This will ensure that fertilizations can be conducted quickly once your oocytes are expelled. Sperm concentration. So ensuring that the concentration of sperm applied to each subclutch is standardized is of paramount importance because of the relationship between sperm concentration and fertilization success. Typically, fertilizations are achieved by sperm concentrations ranging from 10 to the 4 to 10 to the 6 sperm per mil, with terrestrial and foam nesting species generally exhibiting lower optimal sperm concentrations compared with aquatically breeding species. A fertilization curve like the one pictured here should be established through preliminary experiments in order to determine the sperm concentration required to reach an optimal rate of fertilization for your species. Once sperm suspensions have been collected for use in your fertilization crosses, the next step is to determine the sperm concentration within each of your suspensions using a hemocytometer. Counts should be replicated and averaged for accuracy and then entered into an Excel spreadsheet. The spreadsheet can be set up to calculate the total sperm in each of your suspensions, and then based on your target sperm concentration per fertilization tray and target volume of fertilization medium, both of which must be held constant, the spreadsheet can then be set up to determine the volume of sperm suspension and the volume of fertilization medium to add to each of your fertilization trays, shown in this mock spreadsheet just here. Now, regardless of whether your species is naturally, terrestrially, or aquatically reproducing, artificial fertilizations should be conducted in a small volume of fertilization medium. This is known as dry fertilization, and this will increase the probability of sperm and egg collisions. This spreadsheet here shows a total fertilization medium volume of 200 microliters. Additional fluid can be added after a few minutes to ensure adequate hydration, or you can flood your embryos according to the species' natural fertilization ecology. It is also important to select and standardize the osmolality of the fertilization medium. The solution osmolality is important in order to activate sperm motility and allow sperm to reach and penetrate the eggs. Anurine artificial fertilizations are typically conducted in fertilization media of very low osmolality, 0 to 25 milliosmol per kilo, as sperm motility is activated in response to hypoosmotic shock. It is important to note, however, that research suggests that fertilization success in terrestrial breeding species is optimized in media of slightly higher osmolalities. Therefore, the natural fertilization ecologies of your targeted species should be considered. And when possible, fertilization curves should be first established to determine the optimal medium osmolality at which to conduct your artificial fertilizations. Now, both fertilizations and the rearing of offspring should be conducted under standardized laboratory conditions so that genotype by environment interactions can be accurately assessed. Following artificial fertilizations, embryos should be carefully separated and reared individually in ventilated containers. Developing embryos should be randomly allocated to a position on the shelf, and throughout development should be regularly rotated through shelf positions to avoid microenvironmental differences, for example, differences in temperature or humidity within the room. Individual rearing is also essential to avoid introducing density-dependent effects or interclutch competition that can arise when families are raised together. Following these guidelines minimises microenvironmental variations among your subclutches that might otherwise influence estimates of genetic variance. So what fitnessing determining traits should be measured? Well, multiple traits contribute to total fitness. Therefore, you should aim to measure a suite of traits that represent different components of fitness. The first trait to quantify is the proportion of eggs that are fertilized, known as fertilization success. Not scoring fertilization success is a mistake because you won't be able to accurately estimate your early embryo mortality. Genetic incompatibilities that reduce offspring viability are expected to manifest and compound during early development. Therefore, it is important to quantify the survival and abnormalities of embryos, hatching success, and time to hatching. Following hatching of tadpoles from the egg capsule, important fitness traits to measure include larval survival, deformity, growth rate, and morphology. Next, metamorphosis is a physiologically demanding transformation, and it is typically accompanied by a spike in mortality. And so survival to metamorphosis should be measured, along with time to metamorphosis and the size at metamorphosis. In the adult life stage, adult survival, body size, female fecundity, and male fertility are all important indicators of fitness. Finally, assays of performance, for example, predatory escape, exercise and foraging behavior, general movement behavior, and sexual display can all provide estimates of individual fitness and can be quantified throughout larval and adult life stages. Okay, so that sums up the most important practical considerations to cogitate when designing your experiments. I'd like to move on now to a brief case study. The case study I'll be presenting today is a research study that Phillip and I conducted in the brown toadlet, Pseudophrony bibroni. Brown toadlets were once widely distributed along the eastern seaboard of Australia, but in recent years have suffered rapid declines and are currently listed by the IUCN as near threatened. As Phillip has already discussed in significant detail, population mixing or population augmentation is a widely used conservation tool aimed at enhancing genetic diversity. However, mixing individuals from evolutionary distinct populations can have negative genetic consequences, and it's important to quantify the outcomes of population mixing in a controlled setting before proceeding with population augmentation as a management strategy. Australian brown toadlets have been shown to have extreme site fidelity, with males returning to within centimetres of their previous nest site year after year, and so populations are likely to exhibit some level of genetic divergence. With the goal of exploring the potential outcomes of population mixing, we used artificial fertilisation and a cross-classified breeding design known as a North Carolina Type 2 split clutch factorial mating design to conduct a preliminary investigation into the risk of outbreeding depression. Our study involved toadlets from 3 source populations, and fertilizations were conducted in 3 experimental blocks. Within each block, there were 5 sires and 4 to 5 dams, and they were from either Bream Beach or Hyams Beach populations in order to investigate the within population variation in genetic compatibility. And then there was a single dam from the Swan Lake population located approximately 12 to 14 kilometres away from the other two populations. The final combined design involved 85 families of paternal and maternal half siblings, with 70 within population crosses and 15 hybrid crosses. The main finding from the study was that outbreeding consistently had a negative effect on offspring fitness. Mean fertilisation success was very high, 89% for within population crosses. But the fertilisation success was significantly lower at 62% for the hybrid crosses. Both embryonic and larval abnormalities were very common for the hybrid crosses, and only 20% of offspring from hybrid crosses hatched, compared to a high 81% of the offspring that were produced from the within population crosses. Overall, more than half of the hybrid crosses completely failed to produce viable offspring, indicating that brown toadlets are highly susceptible to genetic incompatibility and outbreeding depression. Moreover, because populations were less than 20 kilometres apart, the study provided novel evidence that outbreeding depression in anurans can occur over very narrow geographic distances. We are currently planning experiments to replicate this study on a larger scale and with crosses between a greater number of populations, as well as using a slightly more sophisticated design, as Phil outlined earlier. But this preliminary study does act as a cautionary tale to those conducting amphibian translocations, and it highlights the value of artificial fertilisation to test for genetic compatibility. So in summary, while artificial fertilisation has been used to propagate a growing number of anuran species, AF is yet to be well integrated into amphibian genetic management on a broader scale. We have highlighted that artificial fertilisation is a powerful tool enabling controlled breeding experiments that provide fundamental insights into the genetic underpinnings of viability and fitness-related traits. We also discussed how application of this knowledge promises to greatly improve the implementation and operation of population augmentation for amphibian conservation. While the mixing of populations has enormous potential to aid in the genetic rescue of small inbred populations, widespread implementation has been held back by concerns that mixing populations could cause outbreeding depression. Data obtained from breeding experiments can reveal whether A, there is strong evidence for genetic incompatibility or outbreeding depression between populations, B, whether mixing your populations does not appear to have major detrimental effects, or C, whether there is evidence for heterotic effects or hybrid vigour. Concerns over the potential for outbreeding depression for a species is likely to vary enormously due to differences in the species' capacity for long-distance dispersal and the extent of uniformity and connectivity of habitats in which they inhabit. Now, expecting high species specificity, we encourage conservation practitioners to use artificial fertilisation to conduct split clutch breeding experiments as a tool to inform their decision-making surrounding the use of population augmentation. We propose that by employing artificial fertilizations to facilitate more precise genetic management of threatened species, that this will stand to significantly improve the outcomes of amphibian conservation programs globally. Please find here a list of some articles for further reading on the topic of genetic management. And I will finish up there. Thank you all for listening. And it is our absolute pleasure to conclude the final webinar in this series, which has been on reproductive technologies for amphibian conservation. Thank you, everyone.